Let's now deal with the anatomy and clinical neurology of arguably the simplest part of the central nervous system, and this is the spinal cord. But as we'll see, the spinal cord is going to be the location where we talk about three fundamental neural systems that have neural components found throughout the CNS that begin or end at spinal cord levels. First off, the fundamental anatomy of the spinal cord. It's housed in the vertebral canal. It begins at the caudal end of the brainstem near the foramen magnum with the medulla, and it ends basically in postnatal life at the level of the second lumbar vertebra. And we know well that there are 31 pairs of spinal nerves that arise segmentally from the cord, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and a single coccygeal pair of spinal nerves. And in terms of the spinal cord itself, as is the case for all of the central nervous system, there's a very precise histological organization. We're going to have areas of gray matter and areas of white matter in all parts of the CNS. In the spinal cord, the gray matter is centrally located and is shaped like a funny-looking H or a butterfly. And gray matter, not only in the spinal cord but everywhere else in the CNS, is where we're going to find all of the neuronal cell bodies of any neuron that's located in the spinal cord, along with their dendrites and the proximal parts of their axons. White matter in the spinal cord will surround the gray matter on all sides. White matter is going to be tremendously important because it contains bundles of functionally important specific axons that form a series of tracts of axons or fasciculi, and these axons fundamentally will either go up or down various parts of the spinal cord or ascend or descend its entire length. Let's now look at the fundamental anatomy of the spinal cord and the roots of a typical spinal nerve. The section that's shown on this particular diagram could be from anywhere in the spinal cord, and what you can see is the arrangement of gray matter forming the interior of the spinal cord in the form of a butterfly or an H, and you can see that the gray matter is surrounded entirely by white matter. Note that, again, the gray matter is going to be the location of any neuron cell bodies and dendrites of any spinal cord neuron, whereas the white matter is going to be the location of where we find bundles of axons in the form of a tract or a fasciculus. Note also that we have 31 pairs of spinal nerves, as we illustrated previously, but also note that every branch of every spinal nerve is mixed in that it contains both motor and sensory fibers. And we know from gross anatomy that all spinal nerves branch into a mixed dorsal ramus and a mixed ventral ramus. But here's the additional point. Since all of the branches of a spinal nerve are mixed, every spinal nerve must have a sensory root and every spinal nerve must have a motor root. The sensory root of a spinal nerve is the dorsal root. Its central process of its axons enter at the dorsolateral point of the spinal cord. And since all dorsal roots are derived from neural crest cells, their cell bodies are logically found in a ganglion outside the CNS, in this case in the dorsal root ganglion. The cell bodies of all ventral roots are derived from the neural tube. Therefore, their neuronal cell bodies are found inside a specific part of the gray matter of the spinal cord, and it is only their axons that are coursing in a ventral root on their way to branching and forming part of a dorsal or a ventral ramus. So let's look in a little more detail at the organization of the gray matter, because in addition to forming the locations of neurons, of any neuron in the spinal cord, it has some functional specificity in that the dorsal horn of the gray matter contains sensory neurons, or neurons that are going to respond to sensory input provided to them by the incoming dorsal root. The ventral horn is going to contain motor neurons, in this case, the motor neurons whose axons leave in the ventral root. The intermediate zone, which is part way between the dorsal horn and the ventral horn, is a combination of motor and sensory neurons and includes preganglionic autonomic neurons in spinal cord segments from T1 to L2 and S234, and also contains some sensory neurons in what is known as Clark's nucleus, which is also found from T1 to L2. So the intermediate zone we cannot call either motor or sensory because its neurons have functional specificity for either the autonomic nervous system or some cerebellar type sensation. The next slide summarizes in chart form the basic anatomy of the spinal cord. Note the difference in the corresponding terms. The conus medullaris is the tapered distal end of the spinal cord that contains sacral and the coccygeal spinal cord segments, and in adults ends about the level of the L2 vertebra. The cauda equina represents the dorsal and the ventral roots of lumbosacral and coccygeal spinal nerves that descend below the conus medullaris on their way to exit out of their proper intervertebral foramen. The filum terminale is technically a component of the cauda equina. It is a peel extension that extends below the cauda equina that tethers the spinal cord basically to about the level of the S2 vertebra at the bottom of the vertebral column. And again, dorsal root ganglia contain the cell bodies of primary sensory neurons. Of all primary sensory neurons, they're neural crest cell derivatives. All spinal nerves are going to have a dorsal and a ventral root, making up obviously a pair of roots that join together to form a spinal nerve. As we indicated, we were more specific about it previously. The dorsal horn contained neurons that respond to sensation. The ventral horn contains motor neurons. And the intermediate zone, although not listed here, is a combination of autonomic and sensory neurons between the two horns. And then we're going to use these criteria to help us identify where we are in a particular spinal cord segment. Note that the area of the cervical enlargement and the lumbar enlargement are areas where we're going to find many more motor neurons, particularly, and a larger number of sensory neurons, because these regions of the spinal cord are the segments that provide the innervation to an upper limb or an innervation to a lower limb. So there are going to be some anatomic differences in these regions of the spinal cord, which will help us localize where we are. The cervical enlargement corresponds to the origins of the brachial plexus, and that is the C5 through T1 spinal cord segments, where the lumbar enlargement corresponds to lumbosacral spinal nerve segments from L2 to S3 that's providing a corresponding innervation to the lower limb. And then kind of a 3D kind of picture illustrates, again, the arrangement and anatomy of the spinal cord. You could see, again, the features of the gray matter and how it's divided into dorsal or posterior gray horn, anterior or ventral corresponding horns for motor, the intermediate or lateral horn, the home of autonomic neurons and some sensory neurons. And you could see how the rootlets come out of each corresponding segment, ventral and dorsal, combined to form a spinal nerve that then ultimately branches into its corresponding rami. All right, so now let's get specific by looking first at the anatomic differences between the functionality of various kinds of dorsal roots versus the various kinds of ventral roots, because they obviously have very different functions, and in addition, they have different sites of termination and different degrees of myelination. What we're seeing here is the anatomic differences in the dorsal roots. Note that all of their cell bodies are certainly in dorsal root ganglia because they're neural crests, but note there's a very distinct differences in their sites of termination in the spinal cord and their functionality. Note that we subdivide a dorsal root into a medial and a lateral division. The medial division of the dorsal root contained the heavily myelinated, fastest conducting dorsal root fibers. And these have a classification scheme, basically, that's either based on Roman numerals if they're innervating muscle, or an alphabetic scheme if they're innervating receptors in skin. So note that the largest, most heavily myelinated dorsal root fibers are those that mediate the sensation of proprioception, muscle position sense. And there are two classes of these. 1A fibers are innervating what we call muscle spindles. Those are muscle stretch receptors. 1B fibers are going to innervate Golgi tendon organs. Both are different kinds of receptors that are basically incorporated into skeletal muscle. In addition, the other component modality fiber that is in a dorsal root are the fibers that mediate the corresponding touch modalities of pressure sense, vibratory sense, discriminative touch, light touch. And these are known as A-beta fibers because they're largely innervating cutaneous receptors. Note that all of these dorsal roots that are associated with the medial division enter and kind of sweep medial to most of the dorsal horn because their major destination, in addition to participating in reflexes, is to send collaterals into one of our neural systems, which will form the dorsal columns. So it's really, in a major way, the proprioceptive fibers that are entering also into the intermediate zone or down into the ventral horn to participate in various reflexes. In contrast, note the difference with regard to the anatomy of the lateral division of the dorsal root. These fibers are thinly myelinated. They're known as A delta fibers, or unmyelinated. They're known as C fibers. These lateral division fibers are basically entering and synapsing mainly in the dorsal horn of the spinal cord in various laminae, and they are responding to the modalities of sharp pain, dull pain, cold, and warmth. The sharp pain and cold modalities are conveyed by the A delta fibers. The sensations of dull pain and warmth are being conveyed largely by the unmyelinated C fibers. But again, very different sites of termination with regard to after their point of entry into the spinal cord. All right, and then a final comment. Early on, we subdivided the gray matter of the dorsal horn of the gray matter of the spinal cord into a dorsal horn, a ventral horn, and an intermediate zone. Now, being a little more specific, we can organize these regions of the gray matter into a series of what are known as Rexed laminae. The details aren't particularly important, just to the general terms. For example, the dorsal horn corresponds to Rexed laminae 1 through 6, and in a major way, what's synapsing in the Rexed laminae 1 through 6, parts of those laminae, will be the incoming lateral division fibers of the dorsal root. The ventral horn contains Rexed laminae mainly 8 and 9. 9 is going to be more important because it will contain the cell bodies of axons that exit in a ventral root. The intermediate zone containing autonomic neurons and cerebellar sensory neurons is lamina 7. Now, by contrast, let's do the same thing by looking at the anatomy of a ventral root. The ventral roots largely arise from lamina 9 in the ventral horn, as we just indicated. And certainly the goal of a ventral root motor neuron, as we'll see, is to go out and innervate skeletal muscle. But note that there are actually two kinds of ventral root axons. The alpha motor neurons, as we'll see, will be the axons that exit in a ventral root and go out and innervate skeletal muscle at the neuromuscular junction and make it contract. Gamma motor neurons are much smaller diameter axons. Their cell bodies are still found in the ventral horn, mainly in lamina 9. Gamma motor neuron axons go out and innervate muscle stretch receptors, muscle spindles, and alter the sensitivity of that muscle spindle to muscle stretch. The other point that we want to make about the ventral horn is that the motor neurons in the ventral horn are not randomly situated. They are situated based on whether they innervate flexors or extensors, or whether they innervate proximal or distal musculature, particularly in an area of the spinal cord that's serving a limb. So on the left side of this diagram in the spinal cord, you see how the motor neurons are organized in the ventral horn, in that motor neurons that innervate flexors, particularly in the upper limb, are situated more dorsally. Motor neurons that innervate extensors are situated more ventrally. And also you can see that there is a proximal to distal gradient of innervation across the ventral horn, where proximal motor neurons that innervate trunk muscles are situated more medially. Motor neurons that innervate distal musculature, particularly in the upper limb involving hand muscles, are situated much more laterally. And again, as we indicated earlier, since we need many more lower motor neurons to innervate all the muscles of a limb, this is why in spinal cord segments from C5 to T1, we have what's known as an enlargement, a much larger lateral extension of the ventral horn.